Mark, the floor is yours.
Oh, excellent. And thanks, Janine. And really nice to hear from everybody there and Wendy as well. And I did, I do give Janine an awesome amount of credit for that boost of encouragement that got me across my master capstone. And I'll echo Hank's endorsement of the program. And I'd really encourage folks to check it out. It's got a lot to offer, and it's built had a lot of influence on me as well. So that said, I have a presentation I'd love to share. And Janine, if you don't mind, or Hank, if I just get a verbal confirmation once I'm sharing my screen that you're looking at what you should, that'd be helpful to me. And I think I got that there. Could I get a verbal yes or no?
Yes,
You see the
I
screen
see
now?
the screen. Yes,
Excellent.
Mark, it's there.
Great. All right. Well, welcome. And again, my name's Mark Carlson. I am a master beekeeper, and I'm in San Francisco. And this is my talk on native bees and a bit on nutrition. I've listed a few of my affiliations here on the screen. Some of them I've worked for, some of them I'm currently working for, but the rest of them are volunteers. And we had a quick mention in the early moments before the meeting about how these operations are all based on volunteer hours. And we believe that in San Francisco. We know that's important in Davis, and we sure know that's important with Humboldt folks as too, Humboldt folk beekeepers as well. And I am excited for the bee the bee exchange, the bee social hour on Saturday. I'm really looking forward to seeing a few folks in person. So that said, this is the outline of the material I wanted to cover tonight. The first two chapters, the first two sections are review. They're going to kind of build a foundation by which I'm going to present 3 case studies of work that I've done in the Bay Area for over the last couple of years. And then I want to wrap that all together into kind of an opinion about how there's an opportunity for beekeepers and native bee enthusiasts to kind of have a little bit more in common, and also an opportunity for beekeepers to be advocates of native bees. I think that's a really important step that we can look forward to. And so I do have a couple bee images. These are photographs I've taken, and I want to give a quick shout out to a close friend and colleague, Kara Chang, here in San Francisco. She's with a group called Bee Cause, and they had access to a beautiful native bee collection. And maybe I'll get to talk to Kevin about his at some point too. But they were really generous. Kara was really generous to share that collection. And you'll see these photographs of these zoomed in bee specimens. And I think that's really important to show these tonight because they allow us that connection, not only with the anatomy, but then also how you might be improving your skills to identify those bees in the field. So I'm going to be speaking a bit about each of the families. On your screen right now is a sweat bee, a Halictidae. And in fact, you see the specimens zoomed in. And then I have the live photos that I just took last week, and they were within 10 feet of a bee yard that I have in an urban farm. And so there is a needle to thread here that there is coexistence between our native bees and our honeybees. And again, this will be a review for most of you, so I'm going to go kind of quickly. What are the things that all bees have in common? And in particular, I want to start with that they're all in the kingdom Animalia. They're all arthropods. They're all insects. They're all Hymenoptera. So that means they have an exoskeleton, segmented body, jointed appendages, not 3, not 4, like the arachnids or millipedes. And then they have 2 pairs of wings and a narrow petiole, not a wide one. And that separates them from other insects like flies and beetles. All of the bees on your screen right now are in the superfamily, the bee superfamily, which has about 7 worldwide different families. But I'm going to talk about 5 of them because those are the ones that we identified that were represented in our collection here in San Francisco. And I'll mention a 6th one as well, which I think I located down in Pinnacles National Park. And so the 5 families that I'm going to be speaking about, I'm going to use the same color and name and number as I go through. And so if you see a green number 1, that's always going to be a reference to the Halictidae, the sweat bee family. Callididae is the purple, the number 2, and that's mast and cellophane bees. Then orange is Apidae, and that's the one that has the honeybee, has the bumblebees, has the silver digger, the Habropoda bee. Very, very cool, very special, but also very diverse family. Andrenidae is pink, and then Megachilidae is the blue. So those are the 5 families that I'll be mentioning. But all of these bees, remember, are fuzzy vegetarians. They're not like the ants or like the wasps that are also social like honeybees. There are a lot of solitary bees, but their honeybees are definitely social like ants and wasps. And so we have some commonalities, but we also have some differences. And bees are all pollinators. They're all fuzzy vegetarians. And so they're using the hairs on their body to transport pollen from the flowers back to their nest. And then another commonality between honeybees and native bees is they have a lot of similar senses. And so we know our honeybees as having the antenna. We know our honeybees as having a mouth part. Different native bees have different length mouth parts. Some are longer, but they all have a proboscis. And that proboscis has the same function, which is to take nectar out of flowers. And so they're all common threads between honeybees and all native bees as well. And then visually, we know that our honeybees have both compound and simple eyes, and those are the ocelli. And I have a photo here of the Halictidae, the sweat bee, the green sweat bee, having a bright red ocelli on that image. And so all native bees also have similar forms of visual senses. And that's really important because that's how all bees are flying from flowers back to their nest. And we know that our honeybees are often graceful, but not always. Sometimes they land on the landing pad and it's like a crash of an accident. And then we see that with our bumblebees are sometimes they're not as graceful as well, but they're all strong flyers. And speaking of the wings, they're all the same type of wings. They're, again, a paired membranous, and they're held together by those little hooks. We are all pretty familiar with the hemuli. And then I also want to mention this image that you're all looking at, this beautiful bee wing on the screen here was a photograph taken from a specimen. It's actually from Indonesia, but it's from a lab in Maryland, the USGSB lab. And if you haven't heard about it, do a quick search after this talk, look them up. They're phenomenal. And they just experienced some major budgetary cutbacks. And I just want to echo the importance of our national and statewide and just agency level researchers and how we all hopefully can offer our support to them, because that lab and the gentleman in charge of it, Sam Droege, he has a worldwide influence, and he's someone who can teach a whole lot of people a whole lot about bees. And I've learned a lot from him, and I really treasure his images as something that's inspiring me in some of my photography. Back to all commonalities. I mentioned the vision, I mentioned the flight. I also want to mention how all bees are pollinators. And so there's a mutual adaptation, coevolution that's occurred between the flowers and the bees, how the bees want to get some food and the flowers want to get some pollinators to help spread their pollen around. And so those two have evolved together. And although there are differences in specifics, that some bees are very generalist and other bees are very specific to a certain small number of different plants, in general, what I want to just echo is that they all are familiar with what I wrote here is the pollinator syndrome, which is how flowers have evolved, co-adapted, mutual co-adaptation with the bees. Again, just to get back to the point, how pollen is really important for all bees. And although they're all collecting it, they do transport it in slightly different ways. And some of them put it on their corbicula. Again, I'm using the honeybee as a great example. And these images, these diagrams are just really beautiful depictions of how the honeybee transports her pollen back to her nest. And different bees have different mechanisms, but all bees are transporting that pollen to provision their young, either for that generation to come out or possibly even a subsequent generation down in a subsequent year. Thanks, Janine, for mentioning my interest in pollen identification. It is a lot of fun for me. I have a recent contact in Penn State. Her name is Michelle Mansfield. It's listed here. And she's doing the DNA analysis for some of my honey and pollen. And she's phenomenal. She's got a lot of great ideas. Anybody who's interested in this should check her out. She will give you the floral sources that are in your honey, or if you collect the pollen from the front pollen trap that I have here on the screen, you can give her that as well, and she'll do the analysis on that. People also are able to do microscopy of the pollen grains, and you can look at the size and the shape. And I have Melifloral is the company that I've worked with a couple times, and I really like them as well. Angelina is a graduate student of one of the grandfathers out of Texas A&M, and so Von Bryant. And so a great reference if anybody's interested in Melissa palynology. One final quick connection about pollen identification. You can do it by the DNA. You can do it by the shape of the pollen. If you look at the chemical makeup, you also can identify a few specific chemical markers, fingerprints that are in the pollen itself. And so they're phytochemicals, they're specific chemicals. And my examples that I have here are eucalyptus, and that one has a terpene, which is called eucalyptol, and it tastes kind of like cough syrup. And so you can taste that in eucalyptus honey. You can also measure it with chromatography. And so you can identify your nectar sources, your floral sources using both the DNA, the microscopy, but also other chemical analysis, because you can actually detect it. Not only can we detect it as humans, but you can also detect it chemically. I should also mention fennel honey, because we have a whole bunch of that in San Francisco, and that has anethol, which is a polyphenol. And that flavor, I mean, we all know anise, we all know black licorice, but if you mix that with honey, and I don't know if you can see it on the bottom corner of the screen, that actually is one of the most highly judged honeys in the United States, is the one that has some fennel in it. And so I might bring some of that up from San Francisco when I come up in the fall. And this is a really busy slide, but I do want to kind of make an emphasis on the information here I believe is really important about health and survival of bees. And if you haven't reviewed this, it's really good. I'm speaking mostly about pollen, not nectar. And so pollen is the source of both the proteins and the lipids. And those are, on occasion, are essential. There's a handful that the bees can't make, and so they have to collect from pollen. And they're essential for not only development, but also reproduction and immune responses. And so these different nutritional components of pollen are just the world to the bees. And I have now got this third tag on story, which is to say it's not just the amino acids, the proteins, and the lipids, but also the phytochemicals have a role in bee nutrition as well. I mentioned a few references on the bottom. I know I'm going through this information pretty quickly, but feel free to, I'll share and talk about some of those. In fact, maybe I'll just really quickly, Dr. Pariah Chakrabarty is one of the references on the bottom, and she talked a lot about the 24-methylene cholesterol as being an essential phytosterol for bees. And then we've also had really recent research look at how the ratio of protein to lipids, if you can dial that in, your bees will grow more. They will grow faster and healthier. And so a lot of these nutrition stories are getting really recent research, and we're learning a lot about it. And what I'd like to think too is that we have this connection. All bees require pollen. And so if we're learning about phytochemicals and pollen or protein and lipids in pollen, then those also have metaphors to our other bee communities, because we know they're foraging on the same flowers or similar pollen sources. So we can say that there are some phytochemicals that are specific to bee health, and they have been measured as being able to extend bee longevity, decrease or increase pathogen tolerance, and improve honeybee microbiome. And that's work that was even done here in Davis, in California, but also in Colorado, and is continuing to grow. That field of research is continuing to grow. And then I also really quickly want to mention how not just pollen, but also the phytochemicals in the resins that plant. Our bees are collecting propolis. They're collecting resins from trees, putting that into the envelope. Other bees are using that as well. We know that to be an immune enhancer. It helps our bees and native bees be more resistant to diseases. And they all have very unique tricks that they're using to get through it. So to review these similarities, we know that all bees are Hymenoptera, and they're all in one of 5 bee families. They use similar senses. They're all fuzzy vegetarians, except for a few exceptions that I'll get to called calyptoparasites. But in general, bees are fuzzy vegetarians. And then they use that pollen to provision their young, but they need that protein, lipid, and phytochemicals for their life stage development, reproduction, immune response, and detoxification of pesticides. So these stories will resonate similar with once I get into some of the other native bee stuff. And also on the screen right now, I do have some of these other terpenes that I've been mentioning, not just for fun, but those are things that all have unique flavors. You can measure them in the lab. we do some of that with our honey analysis as well.
Like
Now,
the screen share has gone away.
uh-oh. I think I clicked on something.
Yeah, the screen share is gone.
Oops.
That's
Can I
back.
get a confirmation? Oh, great. Okay. So we went through this, the similarities. Now I'm going to go through the differences between bee families. What makes a bumblebee different than a honeybee? And so we have a lot of specific differences, and they're going to be about the size, shape, the nest construction, behavior, and sociality. Their diet is different. Then we're also talking about natives versus non-natives, managed versus wild. And again, our bees, our honeybees are non-native, managed. But for example, the bumblebee, the big apidae on the screen here, that is a native bee, and that is a wild bee, but it's also a managed bee. Some people are rearing bumblebees. I don't know about Melipygonus, this blacktail, but some other bumblebees are common for rearing. And so I've mentioned these 5 families, and I'm going to go through them a little bit more each in detail. Again, we have Apidae is the one that has the honeybees, but we also have the sweat bees, the Halictidae, the Callididae, Andrenidae, and Megachilidae. Each one of them are really phenomenal. I think I find them interesting and unique in their own way. To begin, let's talk about their sizes and their shapes. So we can have some that are the size of a grain of rice. All of these on the screen are all in the family Apidae, which is the same as the honeybee. And you see the honeybee on there. And I have the honeybee labeled as a bean. It's about the same size as a bean. Or I keep on holding up my finger because I think a honeybee will fit on my fingernail and a bumblebee will fit on my thumbnail. And so those are the differences in sizes as they get big. As they get small, they can get so tiny. And in fact, I have 2 different carpenter bees here. One is the Sarantina is the tiny carpenter, very common in San Francisco. The other larger, the foothills carpenter, is bigger than an almond. It's bigger than our bumblebees. We also can say that Apidae are known for their long tongues. And then I mentioned earlier kleptoparasites, and that's a unique story in ecology because these are bees that actually go in and kill an egg and lay their own egg in that place. And so we have all of these bees in San Francisco, and I'm going to kind of go through those individually when I get to that section. But sticking with the differences between what makes our bees different, so we can say they're different in size and shape. They're also really, this is unique to the different families. They collect pollen and transport pollen on different parts of their body. And so I have a few gray circles on my screen. I hope you all can see them. And then where that particular family transports its pollen. And so I'm going to start with the Halictidae, the sweat bee. And so if you're looking at that one, I say it's got hairy armpits. And so it collects a lot of its pollen on the upper part of its hind legs. The honeybee and the bumblebee, they collect a lot, and the silver digger bee, they collect a lot of pollen on the lower parts. So they either have a corbicula, which is the basket, or they just have like Ugg boots, like they just have fuzzy boots. And if Kevin has seen a couple of those silver diggers, they have these beautiful back legs, silver and covered in back fur, and they're just phenomenal. And that's their pollen collection mechanism.
I saw a fucking silver bee.
Nice. Thank you. We will get to that, and we'll go into the silver diggers a little bit more in subsequent slides. The Megachilidae is very unique that they have fuzzy abdomens, not legs, but abdomens. And so as you're looking at the bees on the flower, try and remember these different categories because that'll help you recognize and hopefully identify what those different families are. The different nest constructions, I have here that 70% of all bees are ground nesting bees or cavity nesting bees. And so the vast majority are not like our honeybees that live in these large colonies. They're actually quite solitary, and they live in small holes or holes in wood and holes in the ground. And each family has its own preference, although you can see both the Halictidae, the Callididae, the Andrenidae, and even some of the Apidae, they live in the ground. Some of the Calictidae and the Megachilidae, they live in tubes, like wooden tubes or like a stem of a plant that's dried up, and then it's got a hollow pith in the middle. And so that's a common place for Megachilidae and some of the Callididae bees to live in. So this is really helpful, not only when we're talking about the health of our native bees, but also we're talking about forage for our native bees, and we're talking about nest sites and nest conditions, habitat conditions. And I'm going to keep on bringing up the silver digger because it's not actually the one on my screen here, but it looks a lot like that. That's actually a different, I think that's called the fog digger or something like that. But it looks a lot like it, and they have similar nests, and they live in the ground. And so I haven't been able to find the silver digger nest sites, but I know they're here in San Francisco, and we know that they really like loose soil and sandy dune terrain. And so that whole story resonates really well. And I really appreciate that comment, Kevin, and we'll get a little bit more into that. Let me just really quickly review that when you are a solitary bee, you emerge in the spring as an adult, you lay an egg, you build a nest, and then you provision that nest, and then you don't live to see your offspring. That little egg that hatches into a larvae and then it pupates over winter emerges as adult the following spring. And so that's a solitary bee lifestyle. And we see that's the same as what the Halictidae, that's how the sweat bees operate. And then our bumblebees, as we know, they have colonies underground. Interesting about solitary bees, though, is they do have aggregations. They have areas where they actually put their nests near to each other, so they have easier access to mates. And so I got to see a nest earlier. This was actually last year. I was being trained in how to find these nest aggregations, and this was actually a digger bee aggregation on Mount Tam. And you can see it's like that penny. If you can see this image, you see a hole that's drilled into the ground that the little digger bee can dig out. And then it's smaller than a penny, but just enough for the bee to crawl into. There are lots of other examples of nest aggregations, and these are interesting because it is something that connects a lot of solitary bees, but I actually found, digger bees, they're kind of one of my favorite. I found a nest aggregation up in Humboldt County in the Lost Coast when I was hiking with my friend last year. And so I documented this, and we put them on iNaturalist as native bee ground nesting aggregations, and they have to be more than 100 individuals. But so I've documented those in Humboldt. I've also documented native bee aggregation earlier this year in National Park, Pinnacles National Park, just south of us here in the Bay Area. And I think this is actually my example of the sixth family, which is much more common in the desert. It's kind of a desert bee, and it's not very common in the US in general, but it's called the Melitidae. And I have these specimens, and I hope to get that confirmation before long too. But I'm really proud to add a sixth family to my list. Like a birder, I like to keep as track of as many as I found as possible. And for vocabulary, again, we're all beekeepers. We all know that we have drones in our colony. We all know we have workers in our colony, and we all know we have queens in our colony. Turns out that's true of bumblebees as well, but we use a different name for it. It's called gynes. And so if you're a reproductive female, so we know our workers are our non-reproductive females, our drones are our males, and then our queens or our gynes are our reproductive females. And so just kind of an interesting separation between the honeybees and the bumblebees that we all should try and keep in mind as we're thinking about understanding bumblebees a little bit better. We also know those colonies are a lot smaller than our honeybees, and we know that they don't have overwintering like our honeybees can. And then I also want to put one other quick plug in here that I got interested in trying to rear bumblebees, and they do this work in native bee labs in Davis. But you can buy these boxes, and I'm catching honeybee swarms a couple a year, and I've caught a few. People will invite me out to actually try and capture their bumblebee colonies. And so I've captured these colonies, they're orphan colonies, and I've been able to rear them in a box and then either release them or at least rear them for a couple months and see them double in size. And it gives me access not only to be able to observe these different social structures, but it really is a great way by which your skills as a beekeeper becomes very applicable to native bee animal husbandry or just wildlife celebration. And so I mention social insects because a lot of native bees are not, but our bumblebees are. And people are raising bumblebees for pollination as well, greenhouse mostly. So sorry again, I've been going kind of quickly. I wanted to get through this background material mostly because I want to set up for my case studies. And so we talked about the things in common, and then we talked about the things that are different. And so the things in common between all bees, and now how are the bees that we're looking at in the field, how are they different from each other? How are the families different from each other? So I mentioned some are bigger and smaller, different types of hair, different coloration patterns, where they collect pollen on their body. That's really important for you to identify the family. Even in the field, you can identify where that female bee has the pollen on her body. That can give you an idea what type of family of native bee you're looking at. Then I also mentioned this idea of nest construction. Different families have different types of nests. Different aggregations of bees is kind of a cool thing, that solitary bees still can be found in similar areas when they're mating, only for a couple weeks out of the year. So it's really hard to track those down, but really special when you know them. And then this idea of life cycle. What is their sociality? Are they living in groups? Are they living in colonies? Or are they strictly solitary? If you want to do some extra reading, again, I'll share these slides with Janine so you don't have to take any notes, but there are a lot of really great free online downloadable native bee guides. And I have 3 here I want to mention. Gretchen LeBaum, who is a phenomenal native bee professor in San Francisco State University here in San Francisco. Arathi Shashardi, who's now in Mississippi, I believe, and works for USDA. And she wrote a native bee guide when she was in Colorado. I highly recommend that one. And Steve Bachman, who's in Arizona State in Tempe and works with USDA and does a lot of really cool pollen stuff. Robin Thorpe and Gordon Franke, those are kind of the grandfather, Robin Thorpe is just huge footprint out of Davis and worked in the same building as Alina did, and just a great influence on a lot of us for celebrating native bees. And Gordon Franke is here in Berkeley in the Bay Area. 2 really good bee guides on there too. And then I also mentioned Sam Droege as being a really excellent bee photographer and a lab that really needs help and support in USGS. Awesome. All right. So you guys are still with me, I hope. So now I want to get into a few examples of projects that I've worked on personally in the last couple of years. The first one, I mentioned Kara Cheng as a friend and beekeeping colleague. I think she's finishing her, going into her finished her apprentice, going into her journeyman of the California Master Beekeeping. She also is leading this other organization, which has got a lot of great opportunities for people to get their hands on bees in San Francisco. And a couple years ago, one of her colleagues collected they recruited a UC Berkeley student to create a native bee collection in their bee yard and also in an urban farm. And then she shared that collection with me, and I got to take a bunch of pictures of it. So I have the person's name who did the collection. Her name is Jamie Palawek. She also did a lot of the majority of the identification. She's an expert and did a phenomenal job. She did both net catching and also supplementary pan trap. Some of the folks from San Francisco Bee Cause, that crew, also helped with the collection. And then once that collection was all assembled and identified, I put it under my microscope and I was able to get some of these photographs of it. So I'm going to go right into it and say, again, green is the sweat bee, the Halictidae family. And so I've shown you this one species in particular a few times now. The male and the female look quite differently, but they are both bright green. And so in this collection, we actually had quite a few of these green sweat bees. And if you go out and you look in your flowers, now's a great time to see them. Like I said, a week ago, I saw both the male and the female in less than 10 minutes of looking. And they're out there. They're smaller than a honeybee. Look for the pollen to be on their armpit, not on the bottom part of their leg. And again, if you see a green bee, in all likelihood, you're looking at a Halictidae. And then interestingly, though, that wasn't the most common bee that we found in this urban farm in San Francisco. It was another, it's called the tripartite sweat bee. And so it's not quite as bright green, but if you look on, that's the one in the middle of those 3 series, the middle one, and you can see the female has all that pollen on her armpits, up on her belly, but also on the upper parts of her leg. And I mentioned that one in particular because that might have been the most common. That might have been the most abundant bee that was caught in this survey. And so we would say Halictidae had good species diversity, as in species richness. It was quite diverse in the number of species. We had 19 different species, but also quite abundant. We saw a lot of this they're tiny. They're actually really hard to see, the tripartite bee. I actually have it on my screen, but they're pretty small and they take a little practice. With regard to Apidae, Also quite diverse. There was good species richness, again, in this urban farm, downtown, not downtown, but urban San Francisco, in the city of San Francisco proper, surrounded by an urban jungle. These bees have diversity and abundance. And so, and in particular, I love I get to segue into our silver digger. But before I get there, there is one common one called the California digger. It looks quite a bit like these images on the top. You can see that's really a lot what our silver digger looks like, and what this potentially is what you're finding, Kevin, up in the dunes of Humboldt. And so the males have kind of this, clepias is the name of their face. You can see how it's really yellow. The females aren't really that showy, but they have very fuzzy back legs. And so, and again, slight difference. We call these digger bees, but these are the Anthophora, and then the Habropoda are the other ones that we're going to be talking about. I also have on here a longhorn, and another really common, a highly abundant type of bee that we had in this survey was a Serentina. And it's this tiny little kind of, it's on the bottom of your screen here. It's just like, it's a little bit iridescent green, which is kind of confusing with the Halictidae. It's a carpenter bee. The male has a little bit of yellow on its face, but it's really, really abundant. And so if you know what you're looking for, it's really great to say that you have a high species abundance of that particular individual. And so we know we're getting a lot of both species richness and abundance of apidae in downtown San Francisco. And some of them are so easy to identify that I want to say these out loud. Like, one of them is called the longhorn bee, and it's because it has these really giant antennae. And so if you see a bee, it looks kind of like the size of a honeybee, and it has really long antennae. There's a good chance you might be looking at this type of longhorn bee. I also have a Vosnesensky, the big bumblebee. You guys definitely have those in your yards, and bumblebees are kind of easy to tell because of their color. But this is the slide I'm so glad to get to. I wanted to mention I'll start with the expert on the subject. That's Leslie Saul Gershon. She's now retired. She used to work for USDA ARS out of Davis, and she can identify the silver digger better than anyone, I think. Well, anyone that I know of. And one of the quotes that she said, because they started to restore these dunes. All right, so if you guys can see my screen, you see the map, and you see up in the northwest corner is where they've done the dune restoration in Presidio National Park. And there's the headline. You see the National Park Service headline there. Rare bee is restored. And so they're now measuring and seeing these bees in a higher abundance than they've seen before. And they attribute that to the restoration efforts to make that habitat, to restore that habitat. We don't have dunes or money or restoration down in the south eastern corner of the city. It's not the most prestigious corner, but we do have dry, sandy soils on hillsides. And I've identified this Habropoda, this bee, for the last, since 2022 was the first year I saw it. And I see it every year. And I go out and I do a catch and release where I chill the bee down. I document it. I photograph it. I make sure I get the wing venation. So Kevin, if you're watching and you're taking notes, the way that you'll know that you're looking at Habropoda miserabilis, the silver digger, is actually one of the easiest way for me is, in addition to you see it on the screen here, but you also need to take a close look at its wing patterns. It's not that easy to do, but if you get a little practice with it, that's how this expert was able to identify it in my yard, and also how she identifies it up in the Presidio. So again, I'm really glad for the conversation around this bee in particular, because I find it an amazing, cool little bee. And I really like what Leslie said here at the top, is that after this restoration, it shows the resiliency of nature, and provides us with hope about how, as if we are native bee enthusiasts, which I think all of us as honeybees, we can say we're enthusiastic about pollination and pollinators, there's hope for their things to be better. And I'd like to think that the last case study showed that they're also, this fact that we saw such diversity and abundance in not downtown, but urban San Francisco, shows that we already have a good starting point. We're not necessarily in a big vacancy or desert of bees. So room to improve, resilience, and something to be hopeful for. Then I also want to mention another one from, this is the San Francisco urban farm, and how this is one other family, the Megachilidae, which is called the leafcutter, or the osmia. Those are two different types of Megachilidae bees in this family. And they both collect pollen on their abdomen, which you can see in the little gray circle. I also think they're just charming and cool little bees. I'm going to go into a little bit more detail on the middle one. So take a quick snapshot of that middle bee called the Megachile peritata. And because I'm going to be talking about a bee that looks very similar to that, that's also very special. Like I'm celebrating our silver digger, which is in the Apidae, I'm going to be celebrating our leafcutter, which is in the Megachilidae. And so, and then also on the bottom, you see a kleptoparasite. That thing is like a little nightmare. That's an amazing little bee that doesn't have to collect pollen. It's called the I wish I could see it now. It's red-legged cuckoo bee. And it goes around and it knocks the other bees off, the other eggs off their little pollen provision, and lays its egg in its place. And they're not that uncommon. It's actually maybe 10% of the bees that we found in our survey were these kleptoparasites. So I mentioned that not all bees are fuzzy. That's an example, but put that red-legged cuckoo bee in your mind as like, what a nightmare. It's even got little ridges on its abdomen to kind of stab the other larvae in order to get its egg and larvae in the right place. So very scary, very, very, very cool biology and ecology. And then another bee that just should be in your short list of bees to recognize, if you see a bee that's bright blue, and you see these other bees on the other side of my screen now that are kind of greenish and bluish, they're beautiful. And those are called mason bees. They're osmia. They're not that common, but if you see them, it's really a great thing to put them in your memory bank as a special bee. The wool carder, also just really awesome, but kind of interesting here that wool carder is a non-native. So that non-native is actually taking some of the resources that a native species might use. It doesn't have the villainification of the honeybee in some references, but it is an interesting reference that we're trying to be honest with what bees are native. That's a special way to kind of talk about them. But I'd also like to say we're talking about an urban farm. We're talking about densely populated San Francisco. And so I don't think of it as a nature preserve. I don't think of it as a national park. I think of it as pretty much an urban center. And so a lot of people and a lot of mix. And so even these non-native bees, like our honeybees, I think are welcome. So first case study, it's all about resilience. It's all about diversity and abundance that actually look really good in San Francisco. I put up a chart on this slide. I hope y'all can see it. Other cities and how they compare, how do we rank them? And so if you look at that last row of the number of species, all of them have about 5 families. That's pretty normal. I bet Eureka has 5 families, but how many species is kind of an interesting question. And so our urban farm had 66. Presidio this year, they just measured 90 different bees in the Presidio National Park, which is awesome and great. You see how Berkeley ranks and Santa Barbara and some of these other big cities. Again, urban centers that have resilient abundance and diversity. Then I also mentioned Pinnacles National Park is just, I can't avoid it. It's such a unique location to have 450 different native bee species. And so that's an example of a very special habitat. And that's actually the segue to my next section, which is not about Tamalpais, but the national park north of San Francisco. Sorry, not about Pinnacles. It's about Tamalpais National Park north of San Francisco, and how we don't really welcome honeybees in national parks. In fact, we don't put honeybees in national parks. That's just like we don't graze our cattle, or harvest the trees. They're protected, specialized habitat. And so we really want them to be special for native bees. And so I was recruited to go and help sample field sites in around Mount Tam and Muir Woods National Park. And so how do they rank, and how well are we able to find special bees in a nature preserve, in a national park? And so I was doing that through 2 organizations, Golden Gate National Park Conservancy and California Bumblebee Atlas. And again, it required a little bit of training and a permit. And then you go out with a net and you catch the bees and report them back through, for example, with the bumblebees, we were reporting them through what's called Bumblebee Watch. And so I reported 3 different species of bumblebees, all captured on a field site. And basically what I did is I walked around with a net. I swung my net. I captured the bumblebee. I put it on ice cooled them down so I could get a photograph and document what they were. And then I released them. So it's a capture and release, not meant to have any mortality on the specimens that I'm looking at. And that's because we want to celebrate and keep these bees as alive and healthy as possible. You see a quick map and a beautiful higher elevation meadow. And you see the different species that I was looking at. And then also documenting their floral hosts, what flowers they were visiting. So you see two different bees. The Vosnesensky is the yellow-faced. You guys probably have that. Also, the Melanopygus is the black-tailed. You probably also have that. And so what flowers do they like? And they were both on a thistle on this screen. And so, yeah, a really awesome opportunity to capture and monitor native bees in a nature preserve. The other thing I was doing on Mount Tamalpais was looking at ground nesting sites. So how well can you find a ground nesting bee? And it's so easy when you go, so easy. If you can find a flower, you can typically find the forage. You can then find a foraging bee. But how do you find where that bee nest actually goes back to? So that requires a little bit more practice, a little bit more thought. And so what I was doing with this exercise was documenting if I could find a native bee nest, and then if I could actually see the native bee come or leave that nest. Most of the time, I never saw any native bees coming or leaving, but I had a lot of fun looking for bees, again, based on the type of soil, the type of drainage, whether it was south or north facing, clays versus sand, or drilled into wood. All of these are different nest sites for different families, different types of native bees. And okay, so I mentioned the silver digger as my cool urban bee story, very rare, very special. And then I want to mention another very rare, very special leafcutter bee. And so this one is called the gumifera, Trachusa gumifera. It's got kind of not a very good common name. I think it's on here. Just the SF leafcutter, which the woman, the scientist who went out, she knew that bee had not been found for a couple decades. And so she made up an idea that she was going to do a search party. And I was recruited to help find the native bee. I didn't find it, but one of the first days she went out during her search party, she was actually able to identify it. And it's a great story again. I wish I had a headline on here, but if you do a search for that Latin name, you'll see there's a bunch of articles written for National Park and other agencies, and they talk about this special find. And so the Department of Fish and Wildlife has designated this bee as one example of greatest conservation need. It's only known to be in a very small area. And in order to respect and protect and stand in solidarity with these conservation efforts, it's an example of where we wouldn't want any examples of other types of bees, non-native bees in that area. We want it to be as pristine as possible for this one specific bee, and we want to know as much about it as we possibly can. And I really appreciate and respect all the opportunity that I've had to work with Sarah Leon Guerrero. She has taught me a ton about native bee monitoring. And in fact, I really like what she said here, and she just got written up in one of the articles, which was there's so much potential for these rare and sensitive species, but sometimes it takes the effort to getting out there and observing firsthand what's truly happening. And so I like that mantra that we can read about these bees, but going out and observing them should be something that we also hold as evidence for where those bees are. And if you can see something rare and special like this bee, like this gumifera, like this, we call it the T gumifera. If you could see and experience that bee or help protect that bee or help restore habitat for that bee, just like the story with the silver digger, the Habropoda, dune restoration, if you can get a rare bee to be back highly abundant, it's a very special conservation restoration success story. So stay tuned. This is a really cool one, and more to come in the coming year. So to review Mount Tamalpais, get out and look. Go out and find these clubs if you can. California Bumblebee Atlas is a great example. Ground Nesting Bee Survey is a great example. I mentioned the nest aggregations. That's another great example of if you see something, report it, and keep your eye out for the things that are very special and unique, and learn to tell the differences between them, because a lot of bumblebees, they might be very special and unique, but they might look like the ones that are very common. And so it takes a little practice. And I also want to emphasize this idea of respecting special status habitat, because unlike San Francisco urban farms, if we can say that there's a special, I don't know, I'll tell a story soon here about an island that was a national park and they had honeybees on there. And so if you can say that there's a preserve or natural space that's desired for conservation, those are conversations we can have about non-native versus native species. All right, I'm doing really good. I'm going to end here in just a couple minutes, and I hope you all are staying patient. This section is for the beekeepers. So obviously, we're all beekeepers in the room, and some recent research that we just put out through UC Davis and USDA ARS, Agricultural Research Service. And it's about nutrition, but it's also how do you measure colony performance? And so we've all thought about this quite a bit. Again, I know this is kind of busy, and I'm just going to kind of crash you guys through it. But if you want, look the paper up and give it a read, and maybe we can talk about it tomorrow or even on Saturday. I'd love to talk more about this data and these findings. And so in almond pollination, again, we're not talking about native bees. We're talking about managed non-native honeybees in almond pollination. If they have supplementary mustard available to them, the colonies grew larger. They were stronger, both in the number of adult bees, but also in their weight. And these were statistical significant findings. And so what do we want to say about that rough overall conclusion? And I also want to mention quickly that they also did it with sunflower, found similar findings. This is unpublished, but the idea is that if you provide diversity of forage sources, your honeybees are healthier. They are bigger, they are stronger, they have higher performance, and they actually have, in some cases, they actually have reduced number of mites. And that's a great conclusion to say, if we can subsidize our efforts to mitigate mites by nutritional efforts, I think it's a great non-intrusive, non-chemical way to be considering how to keep our bees healthy. And then in particular, too, I want to mention how these analyses, like, so here I'm also looking at, you can measure the type of land use, you can look at the type of bloom, the phenology of the flowers that you're looking at, and also corbicular pollen. I think of these as all relating back to native bees because we are talking about a type of bee, non-native managed bee, but we are talking about a bee that visits a flower, and we're talking about the health and nutrition of that bee. And so those stories, those metaphors might be excellent for how we, again, not extrapolate this information onto native bee management, but just how we can be mutually appreciative of both sides of what native bee enthusiasts are thinking about and honeybees management, beekeepers are thinking about. Because we're always thinking about what pollen they're bringing in and what nectar they're bringing in, because that affects our honey. And that does also affect their balanced nutrition. And the more we can learn about this, the better. And I'm really close to the end here. I do mention the UC Davis Health Hub and this project with Project Apis M and the colony collapse that happened earlier this year. Some of those reports that came out, again, I'm going really quick, but it's only because I want to kind of get to my conclusion thoughts and then give a little bit of room for questions. Big takeaway, get out and enjoy nature. I think of taking some of these bee pictures like fly fishing. You go out and you don't always get a great catch, but sometimes you do, and it's really fun and good to try. And some of these photos, even if they're not the most beautiful, they can get me to a level of family identification. Or for example, with the bumblebees, I can identify the bumblebees with photos. And that's a really fun reason to get out and observe your native pollinators, just like you're observing your honeybees on the flower. Whoa. All right. So I wanted to get to this, and I apologize. I went really fast through a lot of material. I think that this will be part of the conversation that we'll have hopefully tomorrow and also on Saturday. But there's been a lot of talk in some recent publications about how is the diplomacy between honeybees and beekeepers and native bees and native bee enthusiasts. And so first, I want to start off by saying habitat loss is kind of the undisputed largest threat to native bees. And so if we want to use the word threat in reference to honeybees, let's first at least acknowledge that the biggest threat is habitat loss. And so if you have no nest sites and you don't have their flowers, you won't have native bees. But if you have nest sites and you have flowers, odds are you will have native bees. That's what we find here in San Francisco. And even further, if you have nest sites and you have flowers and you have honeybees, we still find native bees both in abundance and diversity, at least in this example here in San Francisco and around the Bay Area. I'm really cautious about, as a scientist, about the word exploitative competition, and I really like how Dr. Diana Foster Cox of USDA ARS in Utah, she describes it really well. Good YouTube on that. If you want to be specific about how you use that term as it relates to honeybees and native bees, I really recommend a comprehensive understanding of, before you use it in a title, before you use it as a journalist, be cautious, because it has a different meaning to a scientist and it has an alarming meaning. And I don't think it's often easily measured. So for example, in the last 2 weeks, I've had a bunch of people already text me this article. The first one of that fine print, Travis et al., pollen exploitation by native bee, feral bees. This is in San Diego, suburban San Diego. And again, I often want to distinguish between native preserves, special habitat, and urban centers. Like, I'm not surprised they found a bunch of honeybees at all, because we have a lot of honeybees in San Francisco. We've got a huge honeybee club and community, and I'm really proud of it. And so careful about where you look. And if you want to find honeybees, if you go looking for them in an urban center, odds are you got backyard beekeepers that are maintaining them. Especially if you want to distinguish them from feral honeybees, which requires some real expertise to do that on the flower, if possible at all, which I kind of have my doubts. Island-wide removal, I mentioned this one. This was a nature preserve on an island in Italy, and they said they removed the honeybees and they got their native bees to come back. I actually measured and monitored digger bee and the restoration of that digger bee. I like that article, but I also think it needs to be use caution about island biogeography is not an example. We're on a peninsula, but we should be cautious about how scientists really define the observations on an island versus the observations on the mainland. Neil Williams, an excellent author. He has very good opinions, and he actually might say there's a lot of adaptability of native bees. If native bees are in a place where they have flowers and they have nest sites and they have honeybees, and the honeybees are taking over the flowers, native bees can actually learn, they can go to other flowers. They're adaptable. They're not as oligolectic is the word, and not as specialist as we think they are. So Neil Williams, a great reference. I mentioned the Project Apis M, and how there was a recent paper out about how viruses, it came out, and they said that hives that have miticide resistant mites are acting like dirty syringe needles. And so they're giving the bees that have these resistant mites more viruses. And so that's a great conclusion. But if you read the details and you read how they made those observations, they actually went in the hive and they picked out individual bees that they thought looked sick. And so if a bee looked like it was walking in a weird way, it got selected for its evaluation of if it had viruses. And I'd say I would use caution, because that's not as much of a control as I think we want to be very clear about when we have these kind of, what do I want to call it? It's politicizing topics. And so be cautious, especially when there's an appetite for people who want to read that this is the silver bullet to our problem. Science often isn't that clean, and experimental design is critical if the observations are going to be high quality. Last comment is about Maya McNeil. She just wrote a book. I'm going to bring some copies up when I visit Janine in Humboldt, and I'll leave those copies for you guys to read. She wrote an article about, again, are honeybees a threat to pollinators? She referenced Marla Spivak, who I think is phenomenal. She also mentioned Bernardo El Nino, who I think is awesome and amazing, and he's taught me a ton about honeybees. And he had a great perspective as someone who's not only done a lot of native bee work, but also a boatload of honeybee work. And so I have her quote here, is just how honeybees and native bees can be considered to have a mutual network of pollination, and one hopes for the same for their human defenders. Can we find a way by which we have mutualistic understanding about what our threats are to our native pollinators and also to our honeybees, and then work together in order to solve those? And so that said, I finally got through. I hope I got a couple minutes for questions. And in general, I wanted to echo that there is a role for good beekeeping coinciding with native flora and pollinators. Make the habitat, check out the undisturbed soil, try and get non-native flowers, avoid pesticides, and keep track of the native bees when you see them, because I think we're all stewards of our natural environment. And at that, I want to thank San Francisco Beekeeping Association. I want to thank Florence Fang is another urban farm where I've been doing a lot of work. And thank Janine, and thank you all for surviving what hopefully was a somewhat not too overwhelming talk. And with that, I'll quickly open up the chat to see if there are any questions. Oh, great. Nothing here. It looks like everything is just about the social.
Well, that was wonderful.
Sorry if I went on a little too quickly. Again, I'll leave you with the slides if you want to share them at some point too.
Mark, I'm going to the chat. There might be a question here.
There is a question that was asked by Bruce. Early on, you were talking about pollen. And the question is, I think the answer is yes, but maybe talk about if the research is being done.
Yep.
Can you see forever chemicals in pollen? So I'm assuming he means PCBs, dioxins, or other DDX type compounds. But not only can you see it, but is anyone actually doing that in the United States?
Yeah, definitely. And then interesting, who would, thanks Mary, and thanks Bruce. So who would have the labs and who would have the resources? And somebody like Neil Williams is probably looking at something like that at UC Davis. You can do it. You can actually put your pollen, like I just sent it off to this Penn State lab and Michelle. And so people have the equipment and the tools. I actually have them in the company that I work for. And I actually look at pesticides in my honey. And then maybe even to get to the core of your question. So if honeybees are visiting the flower and there might be pesticides that are sprayed on the flower, do those pesticides get into the nectar? Do those pesticides get onto the bee? And do the bees then transport them back to the hive? And all of them are kind of different depending on the chemicals that you're looking for. But something like neonicotinoids, they can get into the nectar because they're systemic in the plant. They don't have to just be sprayed. You don't have to just have them applications, but they're actually inherent into the tissue and into the nectaries and into the pollen. So that would be an example of one of these, I'll call them herbicides or pesticides, agrochemicals that are detectable in pollen. Whether or not folks are doing it to then see if it's having a negative impact on their honeybees or on their native bee populations, probably, but not in large numbers, maybe a handful of labs around the United States. If anything, a handful of you have probably analyzed the pesticides of your honey so that you knew that it was pesticide free. And so I've done that because I don't know all of my neighbors here in San Francisco. And so I wanted a blanket pesticide evaluation. And so I was able to get that done. Yeah, I'll think a little bit more about forever chemicals, but what do you think, Mary? What's your opinion of that answer? And have you ever looked into it?
I have not. I did do a little Googling and I saw there's a few studies out there. And it looks like one could look at pollen residue, something like PCB residue in pollen as an indicator of urban pollution, or just how, what is currently going on as far as active pollution, maybe coming from air sources, or I don't even know, but it sounds very interesting. Yeah.
Thanks.
Looks like Chris asked a question in the chat.
Would you be, hey Chris, about pollen substitutes. Oh yeah, excellent question. Pollen substitutes. There are a lot of folks that would give a similar kind of style about nutrition, talking about bee nutrition. Oops.
Mark, you still sharing? There you go.
Oh, sorry, folks. So pollen substitutes, I won't really go into it. Sorry, Chris, but excellent question. A lot of people who talk about bee nutrition make that their agenda. So if I'm going to say, here are the things that I understand about the protein to lipid ratio, or here are a handful of phytochemicals that I think are really important, and here are the ones, here's the research that backs that up, it then immediately segues into commercialization. Can you make that pollen to lipid ratio with those phytochemicals into a product that you can then put in a hive and then measure that as being a good solution for bees that need to have pollen supplements? That said, I know that there's been some good work on it and they've done good. I like comparisons that if you have 4 different types of pollen substitutes and then you have natural pollen as an example, and you compare all of those, 9 times out of 10, we see natural pollen grows bigger colonies, grows healthier bees. But how do those other 9 pollen substitutes compare? I think it's in the weeds a little bit. So again, thanks for the question, Chris, and sorry to be evasive, but I like the work that other people are doing on it. I don't have any experimental experience with it, and I don't often feed it to my colonies because we have such horticultural, we never really have a dearth here in San Francisco because of all the urban agriculture and urban horticulture. And if I could, I'm going to segue really quick to that final one too about how do we test our honey for pesticides. So I work in an analytical lab that has that service for large producers. So if you are importing, I don't know, basil or something into the United States, you have to have the FDA stamp it with an approval that it doesn't have pesticides. So I work in a lab that does that evaluation. So I was able to get my honey on their short list and they ran it for me. It costs usually $100, $150 for the full pesticide suite. But for example, if you go to the Penn State Honeybee Lab, they have that service available, I think for about $100. And I think that sending your samples to the Penn State Lab is a great idea because they have great options right now. But I'm also, back to Mary's question, they are going to have my pollen on file. This is kind of like 23andMe. They're going to have my pollen on file. And as she continues to develop cost-efficient ways to do these other analyses, I've expressed my interest to get right first in line. And so should you want to do pesticide analysis in your honey, she has specific recommendations on how you sample it. And interesting, Mary, that it has to do often not only with the nectar, but the pollen in your honey. So if you collect more, if you filter it, then you don't have as much pollen in your honey. And so non-filtered honey is easier for this lab in Penn State to evaluate. And so if you send it for pesticides, you might also review some of their other options, send one sample, try and get a couple analyses run. And I think she's a really fun person, interesting person to talk to, who's trying to grow her extension off the East Coast into California too. Whoa, over time. What do you say, Hank?
No, Mark, we got time. It's only 7:30. I do have a question for you though. How many bumblebees are there in California? Somebody said there's 4 or there's 5.
Huh.
And any of them listed? I think one of them is listed as endangered, correct?
I believe so, right? Because they had to do some special trick with the Fish and Wildlife that they categorized it as a fish, an endangered fish, although it's an insect. And so it does have that specific regulatory status, which means that you have to have a permit if you are looking for it or you're trying to net it. I think that's a Frank. Maybe Wendy's still on here. I don't know it off the top of my head. I know it's up there. It's up in your neck of the woods. It could be on some of I don't think it's far from where you are. I know it's in Northern California. And Hank, maybe I can even send a quick note to Janine and she can do a follow-up on that question. It's a great one, but
yeah,
I don't have that.
That's one that's listed endangered or threatened. And so I wonder, how would you manage the habitat? Would you have to get a permit to kind of do excavating or something like that? I mean, you know.
And then is that a valuable thing to do? And you might say that identifying this, I call it the T. gumifera, the leafcutter that's very rare. It's now under very strict regulatory. And so how about, or the silver digger? That doesn't have any regulatory influence, but it has money and restoration behind it. And so I think of once you identify a species as being endangered, what are the resources that allow biologists and resource conservation, natural resource managers to either improve that habitat or protect that species. And so that would be true of a fish or of a flower. But I'm Hank, really good question. And maybe I'll send a list of the total number of bumblebee species we have in California, where that endangered one is, and I'm almost certain it's in Humboldt County. I know it's in Northern California, and then what that regulatory status really does. What is the purpose of even having endangered category? Because if it doesn't help that bee out, maybe it's, as you said, it just makes for more paperwork for people that happen to have a bee yard in a place where that bee is, and all of a sudden, can you even keep those two together? It's not likely that those would be mutually exclusive. But you're right, building or construction or demolition or something like that might. Oh, good, somebody got it. Kevin was on that. 4 to 5 species of bumblebees in California. Some of them are so close in appearance that, oh man, it's like you look at the yellow face, the Vosnesensky, which is 80% of the bees we have. There's one that looks almost identical to it, but it has a black face. Whew, it's so hard. And so the differences between them can be very small, but I agree. 4 and 5 almost sounds like a small number because you could have something different in the Sierras as you have down here on the coast. I'm reading a comment from Annie. Rural native bees, arguably less. How specific is it? Yeah, how specific? Excellent question, Annie. How specific is the relationship between native bees and native plants? And I said, if we want to have more native bees, let's plant more native plants. That silver digger is really rare, and I've seen it more often on. So the Leslie, the Dr. Seol Gresham, she's in San Francisco. She said you got to look for the lupin, so a native flower, native bee. Is it oligolectic? Is it specific to that? I actually see more of these silver digger on Echium, purple bottle brush, non-native, kind of terrible invasive. It's all over the place. And the other one I see it on is like lavender, like people's yards that have just lavender in their yard. And so I would answer that question to say, depending on who you ask, you can define a species as being, and different species are different, but as I'll quote Neil Williams, they are also adaptable. And in my experience, my personal experience, I've seen some of the most rare bees visiting some of the invasive flowers. So they've adapted to figure that out, which I find really interesting. But I don't want to say that's a general rule. And in fact, it's actually kind of confusing. And I'm sure native bee people might say we should really use caution about if native bees can be okay on non-native plants. But thank you. Really excellent question. Hank, I'll turn it over to you if you think, I don't know if my chat kind of went down, but I'll let you know if you let me know if you think I should hold up or what's next.
I think if anybody has more questions, we can, do you have any more questions for Mark?
In person.
Excellent. Thank you, Janine. That's a perfect segue. I'd really like to continue the conversation. And again, I hope I didn't mean to be a kind of crash course, a little bit too, a little bit kind of fast with a lot of material, but I really do hope that you folks, if you had something, oh, look at that beautiful, Kevin. Nice one. I don't know why it's not. It's kind of tough to tell from that angle, but thanks for sharing that little silver digger there at the end. And Janine and all, thank you very much. And yes, please, if you have questions, come out to the thing, the social on Saturday. I'll be there. I'd love to talk about anything that I talked about today.
Okay, the Bee Social, Saturday. I'm going to stop the recording